Imagine you're looking at two colorectal tumors under a microscope.
Okay.
Morphologically, structurally, they look absolutely identical to you.
Right.
You sequence their DNA, and the readouts match perfectly. I mean, they share the exact same mutated genes driving the disease.
Which happens a lot, yeah.
Right. So you give both patients the standard chemotherapy. Patient A responds beautifully, goes into remission, completely recovers, but patient B's tumor it completely shrugs off the treatment and becomes this hidden, aggressive killer.
Yeah, and that right there is, it's the ultimate diagnostic blind spot in oncology.
It's terrifying.
It really is. I mean, we've historically operated on this assumption that if the genetic blueprint is the same, well, then the disease behavior should be the same, right?
Right. That makes logical sense.
But clinical reality consistently proves that wrong. And for a long, long time, oncology simply hasn't had the right map to figure out why that happens.
Well, today we are tearing up that old map. We're doing a deep dive into a really paradigm-shifting paper. It was published in Nature Genetics by a research team at Queen's University, Belfast.
A fantastic team, by the way.
Yeah, and they've cracked this exact mystery in colorectal cancer, or CRC, by fundamentally changing how we classify tumors. Like, they are moving away from looking at single mutated genes and instead mapping out what they call a pathway-derived subtypes.
Yeah, or PDS.
Right, PDS.
Yeah.
Okay, let's unpack this, because to really appreciate how radical this new map is for you listening, we need to understand the limitations of the old one we've been using.
So the old map is based on something called consensus molecular subtypes.
CMS, right.
Exactly, CMS. For years, this was the absolute gold standard. It categorized CRC tumors based on gene-level expression data, dividing them into basically 4 main buckets.
Okay, 4 buckets.
But the issue has always been the largest of those buckets, which is called CMS2.
CMS2. These are the classic epithelial-rich tumors, right?
Right. And on paper, CMS2 looks like this massive monolith.
Right, like they're all exactly the same.
Genetically and visually, yeah, CMS2 tumors appear uniformly identical. But inside the clinic, patients falling into that specific bucket were experiencing just wildly different outcomes.
Like our patient A and patient B from earlier?
Exactly. So the Belfast researchers realized that looking at individual genes just wasn't giving them the resolution they needed. So they used a bioinformatics approach called SSGSEA.
Okay, wait. SSGSEA. Let me make sure I have this right. Single sample gene set enrichment analysis.
You got it. It's a mouthful, I know.
It really is. So rather than just scanning for a single broken gene, this tool is looking at entire networks. But, I mean, the paper mentions they analyzed over 640,000 combinations of biological pathways.
Yeah, 640,000. It's massive.
Using databases like KEGG and Reactome, how does a tool actually process that much data without just generating pure statistical noise?
Well, it does it by looking for what we call coordinate upregulation.
Coordinate upregulation, meaning what exactly?
Yeah. Meaning, instead of asking if one specific gene is turned on, SSGSEA evaluates if an entire functional network of genes is working together. Like, is it happening in a statistically significant way?
Oh, I see. So it's analyzing the signaling cascades, the actual machinery.
Exactly. And they initially focused their analysis on tumors carrying a very specific driver mutation called KRAS.
KRAS, right, the KRAS mutation.
Yeah. And by shifting the lens from individual genes to these massive pathway networks, they realized that the giant CMS2 monolith, it wasn't a solid block at all.
It was hiding things.
Right. It was actually fracturing into distinctly different biological states.
You know, it's like we've been trying to evaluate the health of a massive forest by running around and just counting individual pine needles.
That's a great analogy, actually.
Right. Like, we find a lot of brown needles, so we just classify it as a brown needle forest. But that gene-level approach doesn't tell us why they are brown. This pathway method is zooming out to look at the entire water cycle, the soil microbiome, the whole ecosystem.
Exactly. You're looking at the forest dynamics, not just the trees.
But let me push back on the premise here for a second.
Sure, go for it.
If you have a broken KRAS gene, doesn't that mutation dictate the downstream effect? Like, if the genetic code is the blueprint, why does looking at the pathway change the picture so drastically?
Because the genetic mutation isn't destiny, it's an initiation signal.
An initiation signal.
Yeah. A mutated KRAS or APC gene, these are the classic Vogelstein drivers. It basically acts like a switch stuck in the on position.
Telling the cell to build a tumor.
Right. But how the cell executes that command depends entirely on its environment and its epigenetic state.
So the context matters.
Hugely. Two tumors might have the exact same KRAS mutation, but in one, the cellular context activates a highly inflammatory pathway, while in the other, it triggers a rapid cell division pathway.
So the genetic starting line is identical.
But the biological machinery actually doing the work is completely different.
Got it. So the mutation is the match, but the pathway determines whether that match was dropped into, like, a puddle of water or a puddle of gasoline.
That captures the mechanics perfectly.
Yeah.
The pathway is the environment reacting to the catalyst.
Okay, let's pause and do a quick 5-minute recap for you listening, just to synthesize where we are before we get into the actual new subtypes.
Good idea.
So, bullet 1. We're looking at a shift from gene-level analysis, which lumped a massive amount of CRC tumors into one identical-looking bucket called CMS2.
Right.
Bullet 2. We're moving to pathway-level analysis. By mapping out the entire biological ecosystem using SSGSEA, researchers can see the hidden mechanisms that genetics alone just couldn't reveal.
Exactly.
Which brings us to a question I want to throw to you, the listener. Think about a time you missed the big picture by focusing on one tiny detail. I mean, that's exactly what oncology was doing with CMS2, right?
It really makes you wonder how many other uniform diseases are actually a dozen different diseases hiding under the same genetic disguise.
Yeah, that's a wild thought. So they apply this network analysis to the CMS2 monolith, and it shatters what actually emerges from the data.
3 distinct pathway-derived subtypes, and they name them PDS1, PDS2, and PDS3.
Okay, PDS1, 2, and 3. Let's start with PDS1.
So they each operate with completely different survival strategies. PDS1 is highly proliferative.
Proliferative, meaning it grows fast.
Very fast. When you look at its pathway enrichment, it is dominated by cell cycle activity. We see massive spikes in MYC targets and E2F targets.
MYC and E2F. And those are what exactly?
They're transcription factors that basically drive continuous, unstoppable cellular division.
So to use an image, it behaves like a speeding car, just the accelerator is pinned to the floor.
Exactly. And the paper notes PDS1 is really rich in LGR5 positive stem cells.
LGR5 positive. Okay, those are the canonical stem cells that usually live at the bottom of our intestinal crypts, right? The ones constantly regenerating the gut lining.
Yes, exactly. But in PDS1, those LGR5 positive cells have essentially been hijacked.
Hijacked how?
The tumor is utilizing the gut's natural rapid renewal system to build its own mass. It is a classic hyperactive epithelial tumor.
Okay, so PDS1 is the speeding car, which contrasts sharply with the second fragment they found, PDS2. If PDS1 is a speeding car, PDS2 operates more like an inflamed battlefield.
Yeah, battlefield's a good word for it. PDS2 is fascinating because it's driven less by the cancer cells themselves and more by the tumor microenvironment.
The surrounding tissue.
Right. The pathways lighting up here aren't cell cycle drivers like in PDS1. They are TGF-beta, interferon alpha, and interferon gamma.
Those sound like immune responses.
They are. It's heavily influenced by the stroma and the immune system. And notably, it doesn't rely on those LGR5 positive stem cells we just talked about.
Oh, it doesn't?
No, it utilizes ANXA1 positive stem cells instead.
Okay, wait. Let me make sure I'm wrapping my head around the ANXA1 distinction. Those are the emergency responder stem cells, correct?
That's a good way to put it.
Like, they only typically mobilize when there is severe tissue damage, like if your body is trying to heal a massive ulcer.
Exactly. So PDS2 is essentially a tumor that has weaponized the body's wound healing response.
Wow.
Yeah, it maintains this state of chronic inflammation to fuel its own growth. Biologically, this maps beautifully onto what we know about the immune-heavy and stromal-heavy behaviors we used to see in other totally different classifications.
But here it is, just hiding inside the KRAS mutated CMS2 bucket.
Exactly. Hiding in plain sight.
But here's where it gets really interesting to me. If we look at the clinical outcomes for these patients, basic logic would suggest that PDS1, the speeding car firing on all cylinders with MYC targets and hijacking LGR5 stem cells, that would be the most aggressive and deadly variant.
Right. Seems like the obvious conclusion, yeah. But clinical data proves the exact opposite.
Wait, really?
Yeah. PDS1 actually has a remarkably good prognosis compared to the other subtypes.
Okay, hold on. How does the tumor growing the fastest have the best survival rate? That feels totally backwards.
It's due to the fundamental mechanics of standard chemotherapy.
Okay, explain that.
So drugs like fluorosil, which are the absolute backbone of colorectal cancer treatment, they're antimetabolites.
Antimetabolites.
Right. They are designed to infiltrate and destroy cells that are actively engaged in mitosis cells that are dividing.
Right, because cancer cells divide faster than normal cells.
Exactly. So because PDS1 is dividing so aggressively, its DNA is constantly unwound. It's exposed.
Oh, I see.
Yeah, its extreme speed is actually a massive vulnerability. It practically absorbs the chemotherapy, making it highly susceptible to standard treatments.
Wow. So its greatest strength is actually its fatal flaw.
Exactly.
The chemo is a heat-seeking missile, and PDS1 is running the hottest engine.
That's a great way to visualize it.
Okay, so that brings us to the anomaly, the third fragment of the CMS2 block, PDS3.
Yeah. PDS3 is what makes this paper a true paradigm shift in the field.
The stealth bomber.
Precisely. When the Queen's University team looked at the pathway data for PDS3, they didn't see the massive cell cycle spikes at PDS1, and they didn't see the inflammatory battlefield of PDS2.
So what did they see?
They saw a near-universal repression of classic cancer hallmarks.
Repression, like it was turned off.
Yeah. PDS3 is a slow-cycling tumor existing in this state of extreme transcriptional numbness.
Transcriptional numbness, okay, meaning the cellular machinery is barely reading its own DNA.
Exactly. It isn't dividing rapidly, and it isn't triggering an immune war.
And it lacks both the LGR5 and the ANXA1 stem cells we just talked about, right?
Yeah. Instead, the researchers found the bulk of the tumor was composed of mature, differentiated cells.
Like they look like regular enterocytes and endocrine cells, just normal gut tissue.
Exactly. It is a masterpiece of biological evasion, and this creates a really severe diagnostic paradox.
I can imagine.
Think about how we increasingly rely on AI in pathology right now.
Oh, yeah. Algorithms, standing slides.
Right. So the researchers tested PDS3 against the NCMS algorithm, which is trained to analyze hematoxylin and eosin-stained pathology slides.
H&E slides, the standard pink and purple ones.
Exactly. Now, the AI can identify PDS1 and PDS2 with really high confidence, scoring an AROC of over 0.8.
Which is a very strong predictive score for AI.
But when it hits PDS3,
what happens?
the accuracy plummets to 0.55.
Wait, 0.55? That's barely better than random chance.
It's essentially a coin flip. Morphologically, under the microscope, PDS3 is virtually indistinguishable from healthy, mature gut tissue or standard CMS2 tumors.
That is wild.
Genetically, it has the same KRAS driver mutations. But the AI just can't see it, and historically, neither could we.
So it behaves exactly like a stealth bomber. It flies completely under the radar, looking structurally harmless, avoiding immune detection, and tricking our best algorithms.
Yep.
But I have to ask, if it's not growing fast, how dangerous can it actually be?
Lethal.
Really?
The researchers validated these subtypes against the PDSC3 trial.
Okay, PDSC3, that's a big one.
Yeah, this is level 1 clinical evidence. It's a massive, randomized phase 3 trial for patients with locally advanced disease.
And what did the data show?
The data showed that PDS3 had the absolute worst relapse-free survival rates. It is profoundly more deadly than the fast-growing PDS1.
Wow. Okay, let's do another quick recap for you listening, because that is a lot of dense biology.
Good idea.
So we have 3 subtypes now. PDS1 is the speeding car, highly proliferative, but easily destroyed by chemo because it's moving so fast.
Right.
PDS2 is the inflamed battlefield utilizing inflammation and wound-healing stem cells to grow.
Yep.
And we have PDS3, the stealth bomber, which is slow, quiet, looks completely normal to an AI, yet carries the most devastating prognosis.
That's exactly the landscape.
So here's a question for you to chew on. If a tumor isn't growing fast and it doesn't look abnormal, how do you think it manages to kill? I mean, you have to wonder, if you're looking at this through a microscope and it looks perfectly mature, how does it actually secure its survival?
Right.
How did the researchers even manage to quantify something characterized by its lack of activity?
And that right there was the massive methodological hurdle.
Yeah.
Because PDS3 is so transcriptionally quiet, they couldn't just look for what pathways were turned on.
Because nothing was on.
Exactly. They had to measure the balance of the cellular state itself. To do this, they engineered a novel metric called the Stem Maturation Index.
Or SMI.
Yeah, the SMI.
Let's dig into the mechanics of this SMI, because it seems to be the decoder ring for the entire PDS3 mystery.
It really is. The SMI quantifies a cell's position on what we call the Waddington landscape.
Waddington landscape. Can you visualize that for us?
Imagine a physical hill. At the very top of this hill, you have a marble representing a stem cell.
Okay, marble at the top.
It has high potential, it's driven by MYC targets, and it's constantly dividing. But as that marble rolls down the hill, it differentiates.
It becomes more specialized.
Right, eventually reaching the bottom as a mature, fully formed cell like an enterocyte in the gut. That downward progression is driven by PRC targets.
Okay, PRC, polycomb repressive complex.
Exactly.
We need to pause on this because it's crucial. How does PRC actually force that marble down the hill? Like, what is the mechanical action happening inside the cell?
Okay, so PRC operates through epigenetic silencing.
Epigenetic silencing.
Yeah, it's a protein complex that essentially alters how your DNA is packaged. You know how your DNA is wrapped around proteins called histones?
Right, like thread around a spool.
Perfect analogy. So PRC comes in specifically through a catalytic subunit called EZH2, and it adds methyl groups to those histones.
EZH2 adds the methyl groups.
Yes, and this causes the DNA to tightly coil up, making it physically impossible for the cell's transcription machinery to read those genes.
It locks them away.
PRC actively shuts down the stem cell genes. It applies the brakes to the cell cycle and forces the cell into a mature, differentiated state.
Okay, I think I have the visual now. So MYC targets are pushing the marble up the hill to keep it young and dividing, and PRC targets are tightly coiling the DNA to force the marble down the hill into maturity.
You've got it.
And the SMI, the stem maturation index, is basically just calculating the mathematical ratio between those two competing forces.
Precisely. And when they applied the SMI to PDS3 tumors, the scores skewed heavily toward the PRC targets.
Toward the mature end.
Yeah, these tumors have actively pushed themselves all the way down the Waddington landscape. They are forcing themselves into a differentiated, mature state.
But the critical question is why? I mean, why does a tumor whose only biological imperative is to grow and consume actively use PRC to shut down its own growth and act like a boring, mature gut cell?
Because it is the ultimate survival strategy against a hostile environment.
A hostile environment like chemotherapy.
Exactly. By utilizing EZH2 and the polycomb repressive complex to enter this state of transcriptional numbness, PDS3 dramatically lowers its cellular stress response.
It just turns down the volume on everything.
Right. Think about what happens during a round of chemotherapy. We flood the patient's system with cytotoxic drugs.
And the PDS1 cells, the speeding cars, they're caught with their DNA exposed.
Yes, they suffer catastrophic DNA damage, they panic, and they undergo apoptosis. They die.
But the PDS3 cells...
The PDS3 cells have their DNA tightly coiled by PRC. They aren't dividing. They simply absorb the toxic hit, lay low, and play possum.
They play possum.
Yeah. They survive the chemical bombardment because they are essentially in a state of biological hibernation.
Wow.
Then, months or even years later, when the environment is clear of the drugs, they quietly reverse the epigenetic silencing.
They unspool the DNA.
They unspool it, roll back up the hill, and cause a lethal relapse.
They survive by weaponizing their own numbness.
Exactly.
It is incredibly insidious. I mean, they aren't outrunning the chemotherapy. They are outlasting it by literally turning off the lights and locking the doors.
It's a terrifying but brilliant evolutionary mechanism.
The fact that we can now mathematically measure this with the stem maturation index it's a staggering leap forward for diagnostics.
It really is.
And what I find incredibly refreshing about the Belfast team's approach is that they didn't just publish this as like an abstract concept. They translated it into an open science tool called Subtype Explorer.
Yeah, which is a brilliant move for the field.
Tell us a bit about that.
So Subtype Explorer is an R-based algorithmic package they've made freely available.
Anyone can use it.
Anyone. Any researcher anywhere in the world can take their own bulk tumor sequencing or single-cell RNA data, run it through this package, and immediately map it against these pathway-derived subtypes.
That's amazing.
They can completely bypass the AI blind spots and immediately see if they're dealing with the vulnerability of a speeding car or the hidden danger of a stealth bomber.
So what does this all mean? We started this deep dive with the ultimate clinical frustration. 2 tumors, perfectly matched under the microscope, sharing the exact same mutated blueprint, yet leading to entirely different fates.
Right, patient A and patient B.
The breakthrough here is the realization that DNA is not destiny. The genetic mutation is just the spark.
The pathway is the fuel.
Exactly. The biological state of the tumor, its pathway networks, its microenvironment, its epigenetic silencing is the fuel. By shifting our map from single genes to comprehensive pathways, we are finally seeing the full battlefield.
We finally have the right match.
We now know that inside that monolithic CMS2 classification, tumors are utilizing entirely different strategies: speeding, inflaming, or hiding.
And that realization, honestly, it forces a profound and somewhat uncomfortable paradigm shift in how oncology approaches treatment.
Because it challenges the basics.
Completely. For nearly a century, the holy grail of cancer therapeutics has been identifying and destroying fast-growing cells. We look for mitosis, we target the MYC pathways.
We target the speeding cars.
Exactly. But if the deadliest variants, like PDS3, secure their survival precisely by laying low, mimicking mature cells, and utilizing epigenetic silencing to ignore our drugs.
It implies our current weapons might be fundamentally mismatched for the most dangerous targets.
Exactly. It raises a highly provocative question. To truly cure these stealth variants, might we need to completely reverse our approach?
Reverse it how?
Well, instead of trying to suppress their growth, do we need to develop therapies that inhibit EZH2 and the polycomb repressive complex?
So take the brakes off.
Yes. Do we need to intentionally wake these tumors up, strip away their epigenetic armor, and force them back up the Waddington landscape into a vulnerable, rapid growth state, just so they become visible enough for standard chemotherapy to destroy them?
Oh, wow. Forcing a tumor to grow faster just so we can finally kill it, waking the sleeping giant on purpose.
It's radical, but it might be necessary.
That completely flips the foundational logic of oncology on its head, and it's a brilliant, challenging concept for you to mull over today.
It really changes everything.
Understanding these pathway mechanisms isn't just academic. It's the difference between fighting the disease we think is there and fighting the disease that is actually killing the patient.
Absolutely.
Thank you for joining us on this deep dive into the hidden architecture of colorectal cancer. Stay curious, keep questioning the blueprints, and always remember to look past the surface level details. We'll catch you on the next one.